Gastrointestinal Physiology, Motility, the engine driving digestion. Welcome to our podcast, where today we explore the fascinating world of gastrointestinal physiology, focusing specifically on motility, the intricate motor function of the digestive tract. Gastrointestinal motility refers to the rhythmic contractions and relaxations of the smooth muscles that mix and propel the contents of the digestive system. This coordinated process is essential for digestion, absorption, and excretion. Understanding gastrointestinal motility. Motility involves a symphony of interactions between the smooth muscle fibers of the digestive tract, the enteric nervous system, the central nervous system, and a variety of regulatory substances. Its primary functions include mixing, propulsion, grinding, and temporary storage of digestive contents. Mixing. Through segmentation, the contents of the digestive tract are churned and blended with digestive secretions, ensuring thorough exposure to the absorptive surfaces. Propulsion. Peristalsis is the process that moves the contents along the digestive tract, from the mouth toward the anus, at a pace optimal for digestion and absorption. Grinding. Particularly in the stomach, mechanical breakdown of food particles occurs, facilitating further digestion. Reservoir function. Temporary storage of contents is provided in regions such as the stomach and colon. The electrical and mechanical foundations. The basis of motility lies in electrical activity known as slow waves. These are oscillating changes in the membrane potential of smooth muscle cells, generated by specialized pacemaker cells called interstitial cells of Cajal. While slow waves themselves do not cause contractions, they set the maximum frequency at which contractions can occur in each region of the digestive tract. When boosted by neural, hormonal, or chemical signals, such as stretch or the neurotransmitter acetylcholine, these slow waves can reach a threshold, triggering spike potentials. Spike potentials, in turn, directly result in smooth muscle contraction. The frequency of slow waves determines the maximum possible rate of contractions, while the number of spike potentials per slow wave defines the strength of each contraction. Patterns of motility. Motility patterns vary depending on the region of the digestive tract and whether the body is in a fed or fasting state. Propulsive patterns. Peristalsis is a coordinated wave-like contraction that propels the food bolus forward. This mechanism involves a contractile ring behind the bolus, driven by excitatory neurotransmitters, while the region ahead relaxes to receive the bolus. This organized movement is often referred to as the law of the gut and occurs throughout the esophagus, stomach, and intestines. The migrating motor complex serves as the housekeeper of the digestive tract during fasting states, cycling every 90 to 120 minutes. Its role is to sweep residual food, bacteria, and secretions from the stomach and small intestine toward the colon, preventing bacterial overgrowth. This complex is initiated by the hormone motilin. Mixing patterns. Segmentation is the primary mixing pattern in the small intestine during meals. It consists of rhythmic, stationary contractions that chop and mix the chyme with digestive enzymes and bring it into contact with the absorptive mucosal surface. Segmentation does not result in significant forward movement of contents. In the large intestine, hostrations are the dominant mixing movements. Contractions of the circular muscle and the longitudinal muscle, known as Taenia coli, cause the colon to form sacs called haustra. As these haustra form and relax, the contents are rolled and mixed, facilitating absorption of water and electrolytes. A regional journey through the digestive tract. Mouth and esophagus. In the mouth, chewing is a voluntary process that mechanically breaks down food and mixes it with saliva to form a cohesive bolus. Swallowing is a complex reflex with three phases: the oral phase, where the tongue pushes the bolus into the pharynx; the pharyngeal phase, which involves rapid, coordinated movements to propel the bolus into the esophagus while protecting the airway; and the esophageal phase, where peristalsis carries the bolus downward. If food becomes lodged, secondary peristalsis, a local reflex triggered by distension, helps clear the passage. Stomach. The stomach is divided into two functional regions with distinct motility roles. The proximal stomach, including the fundus and body, undergoes receptive and adaptive relaxation, allowing it to accommodate incoming food without a significant rise in pressure. Gentle tonic contractions push contents toward the antrum. In the distal stomach, rhythmic peristaltic waves mix, grind, and propel the contents. The pyloric sphincter opens slightly, permitting only small, liquefied particles to pass into the duodenum, while larger particles are sent back for further grinding. Gastric emptying is carefully regulated by feedback from the duodenum. Optimizing digestion and absorption. Small intestine during meals. Segmentation predominates, ensuring thorough mixing and absorption. Between meals, the migrating motor complex clears the lumen. The ileocecal valve regulates the flow from the ileum to the cecum and prevents backflow, responding to stimulation by gastrin and distension. Large intestine in the colon. Hostrations facilitate slow mixing movements for water and electrolyte absorption. Powerful mass movements, occurring infrequently, propel feces over long distances and are often triggered by the gastrocolic reflex, which increases colonic motility after a meal. Defecation is governed by a spinal reflex. Distension of the rectum initiates relaxation of the internal anal sphincter and voluntary relaxation of the external anal sphincter. Control of motility. The control of motility is governed by a hierarchy of signals. Locally, the myenteric plexus within the enteric nervous system can independently mediate reflexes such as peristalsis and segmentation in response to stimuli like stretch, changes in pH, and osmolarity. The autonomic nervous system exerts influence, with the parasympathetic system generally stimulating motility and the sympathetic system inhibiting it and contracting sphincters. Hormonal signals further modulate motility, with agents such as gastrin and motilin stimulating activity, while secretin, cholecystokinin, and other peptides act as inhibitors. Conclusion. To summarize, gastrointestinal motility represents a beautifully orchestrated sequence of events, transforming a meal from ingestion to expulsion, maximizing nutrient extraction, and maintaining the health of our internal environment. Thank you for joining us on this deep dive into gastrointestinal motility. If you appreciate our content, please remember to like, share, and subscribe for more insightful discussions. This is the Everything Surgery Podcast, where we dissect the extraordinary.